All right, there we go. And I just wanted to say thank you to everyone that took the time to join us today. Really appreciate the attendance. I think we had over 70 people registered, so I know a lot of folks have probably registered just so they get that recording link that might not be able to join us right now, and likely we'll have some others joining us as we get going. But thank you for taking the time. And if you're here, of course, you certainly have interest in the Barley Pest Initiative and are serving as a researcher or a partner, and we really appreciate that support. This initiative is a few years in now, getting a bit more mature, and really excited about the progress that we've been able to make. So to go ahead and get started today, if you were in the forum last year, we actually used the Slido tool to manage doing some polling and also some Q&A. And so if everyone, if you want to participate, would appreciate if you did, pulled up your smartphone, and you can actually also log in on your computer and go to Slido. If you have a smartphone, you can usually pull up your camera and scan that code. It'll take you to a site, or if you do it on your computer, you can just type in that web address there and enter that code, and that'll get you joined in. This is going to allow us to do some interactive polling, and then also if you want to enter questions through that, that would be super helpful. It's a little bit harder to manage questions, especially on Zoom when you're trying to run the meeting as well as monitor the chat. So you can certainly use the chat on Zoom to ask questions, but feel free to enter them on the Slido app as well. And this, I shouldn't say that, it's not even an app. I mean, there is an app, but you don't have to download anything to use this. So hopefully if you scanned that code, you got right to it. So, all right. With that, we will go ahead and get started. And just to test things out, to get our fingers moving. So first of all, I just want to ask about affiliation. This is really just to kind of test to make sure everything's working okay. But on here, you can check all the things that apply and then go ahead and send in your entry just to kind of get a feel for who's in the room today with us. Right, awesome.
This is funny.
Go ahead and keep entering if you want. I'm going to bring some more people in from the waiting room. All right, awesome. So overwhelming majority of you are BPI funded researchers, which is great. You're definitely in the right place. Some additional researchers, possibly not funded through BPI, hopefully collaborating on some of those partnerships though. Also have some members of our National Barley Improvement Committee here, which is great. And some of our administrators. And I guess I should go ahead and also click on the AMBA ones. We've got at least one AMBA person here. All right, great. Well, thank you. Thanks for everyone that participated in that. 32 of you, that is great. All right, so next we will go through, and I'm just going to provide some background on the BPI. Apologies to those of you that have heard this spiel before, but there's some of you that are pretty new to the initiative, and so I just wanted to provide an overview of what that's all about. So striving for better barley, and that is really kind of an overall theme of everything we do at AMBA and the work we do through the National Barley Improvement Committee. But the two main priorities for the BPI are first, to develop genetic resistance and management strategies to mitigate the negative economic impact of over 20 pests. And to put some context there, we're looking at insects and diseases other than fusarium head blight, and we've got over 20 on our priority list. And then secondarily, targeting insect pests, bacterial, viral, and fungal diseases. So that's what's included in those 20 plus pests that we identified. And so that's the work that we do through the BPI. I would say a majority of the funding does go to that genetic resistance work, but we do have a few folks working on the management strategies as well. So BPI by the numbers, primarily from last year, we haven't had a huge change in the number of collaborators. The only thing that I will mention is that this last year we were able to actually secure the funding to also put some support towards grain genes, which I don't have on there because we're still working on getting that funding where it needs to be, but that will certainly be an established piece of this work moving forward to make sure that we have that database in the background helping support and capture the work that you all are doing. We do have a very diverse set of BPI funded research projects. And this past year, it actually yielded 9 new unique publications. And some of those are flagged in the BPI annual report I sent out, but it's not all inclusive. So there's some others that I didn't put in there, but very excited to see new publications and new work coming out. And also saw many of you report that there were going to be new publications likely coming out this year. So that's excellent. And we do have over 50 researchers engaged in this work, working across 15 states, which is excellent. And for any of you that ever done any work with the National Barley Improvement Committee, this diversification is excellent because every time we go into these state offices, we're able to talk about how this research investment is directly impacting their state. Here's a little bit of background on our funding progress. So when this initiative was brought together at the start, the fundraising goal was actually $4 million, and then we added a couple partners along the way. So our target right now is $5.3 million, and that would be an annual appropriation through the Ag Appropriations budget. But if you followed it all, the kind of chaos in DC right now with the budget, we are still sitting at our FY23 level of $3 million. Although for the FY24 budget that would have started last October, there was a tentative $4 million written in for both the House and the Senate proposed budgets. And so we're very hopeful that that $4 million will come to fruition. Would be great if it came to fruition before FY25, but I'm not sure. The next budget round deadline is October 1 for the Ag Appropriations budget. So hopefully we'll have some good news by then, but not holding my breath with as many times we've had to kick the can down the road lately. But very exciting that we're up to that $4 million. And I will just say that when we go to DC in a month from now, we will be asking for that full appropriated amount of $5.3 million. So as you can see on the graph there, we've incrementally increased our appropriation $1 million every year. And so I'm hoping that we can get $1.3 million to push us over the finish line in this final year. So very exciting stuff. We're almost there. And this will also mean more funds to all of your programs. And I just want to say thank you to everyone who took time to fill out the BPI questionnaire. The Barley Pest Initiative annual report is something that we post to the website. We share with kind of our congressional leaders that have helped secure the funding for this. And really, it just provides a nice snapshot to our stakeholders on what we are doing with the investment. I know it can be a bit of a cumbersome process to fill out additional reporting, but I can definitely say that it is very helpful for the work that we do at AMBA and MBIC. So thank you for those of you that did that. So I now want to just highlight a few of the researchers that are doing work on the initiative. And again, because I'm trying to manage the screen and all the Zoom workings behind the scenes, I don't know if our first presenter is on. Juliet Marshall, are you out there? I didn't think I saw her. All right, so we are going to go ahead and jump over her slides onto our next presenter. Do we have Nate on from Ohio State? Yay, there you are.
I'm here.
Awesome. So Nate, I will go ahead and click through the slides for you. So you just tell me when you are ready to progress, okay?
Okay, great. Thank you very much. So I'm going to give a brief overview of some of the work that we've been doing in the Jacobs Lab here in the Department of Plant Pathology at Ohio State University. led by Dr. Jonathan Jacobs, and I'm presenting. I'm Nate Hayden. I'm a PhD candidate in the team. And we have a picture of the team down here. We like to have fun when we're doing research. So this is a picture from a holiday gift exchange here. And I want to say, first of all, thank you to our funders, especially American Malting Barley Association, as well as we've gotten internal funding from Ohio State, as well as from USDA NIFA. Go to the next slide. So our focus is on the causal agent of bacterial leaf streak on barley, which is Xanthomonas translucens. And there's two different pathivars I'll get a little bit more into. One of those is the pathovar translucens, which is very specific to barley. And then there's another one, Angelosa, which is more of a generalist. Sorry, I put a bunch of animations in here.
No worries.
But basically, the main point of this slide is that in the life cycle, you get these water soaking here you see in this image. And one of the things that we're really focused on is what's the diversity of this pathogen, because it's becoming an increasing threat to production on a global scale. And so we're interested in what the diversity is and how it's transmitted. And there are two main mechanisms that have been found and studied for transmission of this organism, Xanthomonas translucens. And one of them is thought to be more of a secondary inoculum and a short distance transport is you get these exudates that you see in this picture forming when you have a high concentration of bacteria, and they ooze out of the leaf, and these actually dry and spread in the wind. And if you've ever seen them, it's very hard to control and stop them from spreading. The other way that these bacterium is thought to spread long distances is through seed transmission. And so there's a lot of regulations right now also about seed transport internationally. It's a major area of concern that can be very expensive for some companies. Go to the next slide, please. So one thing, regardless of how it's being transmitted, Xanthomonas translucens is present all over the globe. It's done a very good job getting around, as you can see here in this image from EPPO. We actually, Jules Bouchakis and I've helped a little bit, he's developed a new fact sheet for the EPPO here, which is a kind of European fact sheet central for different plant pathogens. But you can see all these yellow dots where Xanthomonas translucens at the species level has been found. But barley infecting strains, those two different pathivars that I mentioned briefly, are actually form a specific clade, as I have here in this box, which is different from other pathivars of Xanthomonas translucens. So we see that there is the separation of strains that infect barley specifically. The next slide, please. And so we wanted to understand more what the diversity within this group is. So just going to briefly mention, so we have an ongoing collaboration with Dr. Ruth Mackey's lab at University of Minnesota, as well as Rebecca Kurland in that group. And we found a couple of really interesting things in a recent publication in phytopathology. We found that actually, despite this global spread, there isn't really a geographic bias in terms of where the clusters of strains are. So there's 3 clusters of Xanthomonas translucens, pathovar translucens, that's the barley specific pathovar. And all 3 of those clusters are present globally. So they're present in Iran, where we have a lot of sampling, as well as in North America and in the United States. And so despite this global distribution, we really see that these strains from all different groups are spread out. And the other thing is that we want to look at the effector complement of these strains, because there's been increased breeding efforts to understand, try to locate resistant germplasm. And but we didn't know if you could really use one representative strain, or if you had to use several, and what the diversity of effectors were in this strain group. And we found actually that there is a wide set. Here's type 3 secreted effectors in different strains here from both Asia as well as the US. And we see that there's a really wide set of about 17 effectors that are highly conserved. But if you look at one class of effector, TAL effectors, which bind to host DNA and manipulate gene expression, we see there's actually a huge amount of diversity. And so this leads us to recommend that we really ideally want to try resistant germplasm against multiple strains. Next slide, please. So these could be potential elicitor targets here, but also we see some a lot of variation in the different virulence factors here, these different effectors. And so one thing that we have ongoing, and in collaboration with Dr. Brian Stephenson at also at University of Minnesota, is trying to understand how effective is some of the resistance that he's found going to be against a more diverse Xanthomonas translucens population. And so here I have some really preliminary data. In this first figure, we look at from spray inoculation, percent symptomatic area, and this is on young plants. Typically in the field, they have to use really, adult plants, and it takes a long time, and they're kind of at the mercy of the weather conditions. And we're trying to develop rapid phenotyping techniques so we can screen these different strains against a panel of barley more effectively. And so we have here susceptible check MW14, and then next to it is actually supposed to be resistant line PI 329,000. And then we have Quest, which is actually a diploid variety that's supposed to be quantitatively resistant. And these young plants, we see that Quest, that quantitative resistance seems to show up, but it might not be the resistance that's more that's stronger. It might not be relevant to the young plants of PI 329000. And what was interesting is, okay, we wanted to know, is this going to show up if we inoculate a broad set of strains and not just the one? This is a limited set here. So if you look again, we have the same type of graph, but we have just the susceptible check versus Quest, that quantitative resistant line. And it varies, and we don't have a lot of significance because of the limited size of this trial, but we see preliminary evidence that this might be broad spectrum. So we hope to use this type of method to get at what age are these different resistances relevant, and then can we track them against different barley lines and see if there's risk for them to break. One more slide, please. And then one last thing I just wanted to briefly mention is that Dr. Jacobs and collaborators found that between those two pathivars, there's actually a distinct lifestyle where one is vascular and one is non-vascular, and there's a marker gene for that. And we've developed, Dr. Roman Reina, postdoc and former postdoc in the team, developed that a set of primers that can differentiate between the two different pathivars. And with that, I think I might have gone a little bit over, so I apologize, but I have, there's a few different publications that have come from this work here that I have cited on the next page. Thank you very much for listening.
Yeah, no, thank you for rushing through that. And I really appreciate everyone keeping to time as much as possible, only because I want to give everyone just a brief taste of some of the great work that's happening here. And of course, these presentations aren't meant to be all inclusive. It's more about just letting you know some of the things that's happening through the BPI. And now you can reach out to Nate and the team at Ohio State if you guys have any other questions. We will do some Q&A once we get done with all the presentations. I'll open it up. But just to keep us rolling through these, I will go ahead and go on to the next presentation. Also, feel free to use that Slido app as well if you want to go ahead and enter a question in there. And if you're on there and want to answer your own question, you can do that. And I did see, yeah, Ruth says, good job, Nate. All right, so the next one we're going to go to Dr. Wise. And looks like you're off mute already, so go ahead and you just let me know when you want this the slides progressed, okay, Roger?
Okay, thanks. I'm just going to give a short overview of some of the work we've been doing in the last few years on protein networks as a molecular foundation for next generation resilience breeding. Next slide. So as we know from quantitative genetics, no gene works in a vacuum, and I'm a gene for gene guy and host parasite interactions, and so over the past 20 years, we've learned a lot more about the molecular biology of gene for gene interactions and recognition of host resistance by avirulence effectors. But the question is, what lies underneath? We know there's a whole proteome interacting in response to a lot of stresses: pest, disease, drought, cold, and heat. Okay, so that's the main question. We like I said, we work with gene for gene interactions, but what lies underneath? So next slide. Okay, so we work with powdery mildew infection as a developmental system. So like Nate before, this is basically the life cycle of the asexual stage. where you have a spore lands on a leaf, it sends out episodial germ tube. And within about 24, 30 hours, ostoria begin to form. And in 5 to 7 days, you get conidial stalks shooting up, and they blow off in the wind or are shaked off by some graduate student onto new plants. And the key here is that the powdery mildew only affects epidermal cells, which makes it very nice system to work with. It's an obligate biotroph, though, so you cannot grow it on a plate. So everything that we see is interdependent upon the host proteome as well. So our goal was to make a interactome between host and pathogen. And so we were on an NSF ERICAPS grant a while back, and we got a library of almost all the powdery mildew effectors and synthesized for the group. And we got one copy in our freezer in Ames. And we also developed RNA-seq of this life cycle on our wild type plus a lot of mutants that we use. And from that same RNA prep, we developed a yeast-2 hybrid next generation interaction library. Next slide. So if you skip 10 slides over, that ends up with this result. And so this is the most stringent interactome we've come up with. It's 1,085 proteins, 1,497 interactions. And all these, the red are the effector baits. We use 50 of those, as well as some MLA fragments, which is a major powdery mildew resistance gene. MLA also encodes specificity to stripe rust, rice blast, UG99 stem rust, and spot blotch. So we think that things that we find out here, we can hopefully apply to those other diseases as well. So as you can see, there's a lot of interactions going on. And if we go to the next slide, I'll just show you a sub network with just one kind of interaction. CINAs are ring type E3 ligases that modify host defense. And we probed the library with this one effector called CSEP0219 and pulled out 5 individual CINAs on 5 chromosomes. Well, they're from 5 chromosomes. And and we've confirmed those, and we're working up a publication right now with our collaborators in University of Copenhagen, Hans Sordal Christensen and Karsten Petersen. We also have seen a lot of other interactions, as you can see, and like we said, we have 1,000 to do a deep dive on. And right now, I got to remember to put the call out. All these have addresses in the genome. And so they can correlate to any QTL that anybody else has found. All we have to do is get together and crunch the data and see what test different hypotheses. So I'm just putting that call out that we have all these addresses, and I will be contacting other people that I know in the breeding community to help me out on that project. Next slide. Okay, so this is a summary of the thousands of interactions we found of MLA and AVR MLA. SS 2. Summa scores 2.5 is from our new software that's really high confidence. And so we've found 7 and 12, 4, 1, 5, and 8 new interactors we have to do research on, and that are besides the actual initial recognition of the AVR to the ML to the R gene. And many of these associate genetically these targets with MLLA at penetration or MLA during developmental hysteria. And the final conclusion is that in the barley bloomeria system, these interactions, as you see before, some converge on a common target. Many are unique. Many effectors have somewhere between 3 and 10 targets. So there's a lot going on at all different layers and all different cellular compartments. So that's our job now to figure out what that is. Next slide. And this is just a picture of the group. We're a lot smaller now, but this is the people that did the work. and our funding sources. So thank you.
Awesome. Thank you so much, Roger. Thanks for blowing through that quickly for us.
How did I do for time?
You did very well, very well. I really appreciate kind of this high level, you know, this is really meant just to entice people to reach out, connect, because there's not a lot of opportunities to do that. I certainly hope that at the next Barley Improvement Conference, which will be in 2026, that we will have a designated session that's all about BPI projects. So as these programs become into maturity, I'm really excited that we'll be able to do more expanded presentations then, but appreciate the quick ones right now. So we'll move on to the next one. And we've got Dr. Barman with us from ARS in Lincoln, Nebraska. Are you on? I believe I saw you. If you're talking, I don't hear you. There we go.
Yes.
Okay, great. Go ahead and you just let me know when to progress the slides.
Yes. So good afternoon, everyone. I am working with Dr. Satyanarayana Tatirani. So now let me tell you a short story about barley yellow dwarf virus, their strain composition and distribution of different aphid species in Nebraska. Can you shift in the next slide? So barley yellow dwarf virus causes huge losses to the growers of different cereal crop. As you can see here, the symptoms of barley yellow dwarf virus, that is reddening of the upper portion of the leaves. So we survey different location of Nebraska and collected the symptomatic leaves from the various location, and we standardize one protocol, that's multiplex PCR, that can detect all the strain of this BYDB at a single reaction. Next slide. Yeah, let's look into the interesting finding. What we found, you can see here there are different kind of samples, that is wheat, oat, triticale, and rye. In case of wheat sample, if you look into these wheat samples, so here x-axis representing the different strain of different combination of BYDB strains, and y-axis representing the number of BYDB positive wheat leaves. So you can see here HGB is the dominating strain of BYDB present in the wheat samples. After that, HGB MAB co-infected samples. And the another interesting finding is we got lot of all different strain present in the particular samples of wheat. So in case of oat and triticale and rye, we found the similar kind of strain, that is HGB is the dominating strain in oat, triticale, and rye samples. Next slide. Now let's see which aphid species is prevalent in the particular the dominating aphid species present in the Nebraska. You can see here x-axis representing the different aphid species. You know, the barred cherry wood aphid, greenback, there's English grain aphid which can transmit this barley yellow dwarf virus strains. So here x-axis representing the different aphid species and y-axis representing the number of aphid species. So here we can see the meat, hallam, odel, scribner, stump, everywhere barred cherry wood aphid is dominating aphid species compared to the greenback. So our previous finding means the previous scientists reported that greenback aphid can transmit this BYDB viruses more effectively compared to barred cherry wood aphid. But our finding is contrary with their finding. We are getting more barred cherry wood aphid here. So we have to further confirm that there's a transmission efficiency changes or not in the barred cherry wood aphid or greenback aphid. So we have to work on that particular topic. So next slide. And now to understand the viral load in the different location of Nebraska, a multi-location trial was conducted by using the randomized complete block design. And you can see here we chosen 3 locations, that is meat, scribner, and hallam. Meat, scribner, meat, and there's 3 different other location. One is Jefferson County and another one is Concord. So we already shown the 5 different popular variety of barley and wheat, and we are waiting for upcoming summer for the data from that particular field. And before thank you to everyone, I acknowledge Dr. Satyanarayan Tatinani, Dr. Soundpias Mondal from our lab, and my collaborators and wheat and barley growers from Nebraska and Kansas State, and my funding agency also. Thank you.
Excellent. Well, thank you very, very much.
Yeah.
All right. So I haven't seen Juliet hop on yet. I even sent her a text. I wonder if she got caught busy with something else. So we will go ahead and go to our last presentation now, which is going to be Eric. Yeah, you're on. Perfect. Just let me know when you want me to progress.
Hi, good afternoon, everyone. I'm Eric Nazarino. I'm a postdoc at the University of Minnesota. I'm working with Brian Stephenson and also Matt Rouse at the USDA Zero Disease Lab. So in the past 2 years, we have received materials from several barley breeding programs across the United States, including Anheuser-Busch, University of Minnesota, University of Nebraska, Oregon State, Ohio State, and Virginia Tech. So we did a screening for about 870 lines in the past 2 years for barley leaf rust. And we do this at the seedling stage, as you see in the picture. So what we do is that we screen them against a moderately avirulent isolate of the pathogen. So a mild strain of barley leaf rust. And then whichever lines are resistant to that isolate will be moved to the next stage where we do gene postulations. So we use additional 3 isolates to postulate for genes such as RPH 3 or 9, RPH 5 or 6, and RPH 7. So next slide, please. This is a poster that I have presented at the Barley Improvement Conference a few weeks ago in San Diego. So it says that only up to 51% of the US barley breeding germplasm are resistant to leaf rust. So that's based on our 2-year data. And here you can see all the 6 breeding programs. And per year it's color coded. It's blue for the 2002 and red for I mean green for 2023. So those are the percentages of resistant lines per breeding program. And I also included here the postulated genes that were identified in the breeding germplasm. So as you can see here, only Minnesota and Virginia Tech generally has a moderate level of resistance to barley leaf rust. And I guess that's one of the goals of this program is to enhance the resistance in other barley breeding programs in the US in the future. In our group as well, we not only study the host side, we also study the pathogen side. And so we do this annual surveys of Puccinia horrii, and across a 30-year period, we found that there's a variation in virulence within the United States. And so that means that resistance genes in the West Coast may not be effective in the East Coast and vice versa. And so if you wanna know which genes are still effective in your region, you can check the QR code, which contains the link to a recently published paper about the virulence dynamics of Puccinia horrii here in the United States. Next slide, please. So in the past 2 years, we have received materials from 6 breeding programs, but I'm happy to announce this year that we've got additional alliance from 3 universities, including Montana State University, Cornell, and UC Davis. So I'm happy to expand this service for other breeding programs out there. And so if you have any questions or if you want to join or send seeds to us in the future, please don't hesitate to contact me. Yeah, thanks Amba, and thanks to the Barley Pest Initiative for funding our research.
Great. Well, thank you very much for presenting today. And we do have some time, especially since we didn't get Juliet's presentation. I'm going to actually stop sharing my screen just so you do have your camera on. We can see each other all a little bit better. I guess I just wanted to open it up. If anyone had questions for the 4 presenters today, or just wanted to reach out to connect on anything, feel free. We've got a few minutes where we can do that right now. So any questions?
I have a question for Eric.
Mhm. Yeah, go for it, Eric.
The genes that you're seeing resistance in my germplasm, a lot of them are shared with the Anheuser-Busch program, and a lot of my lines were crossed to their material. So I'm wondering, are you screening their spring material or winter material?
I haven't looked specifically on the information about the
Okay.
line. So
Okay.
I can take a look and let you know.
Okay.
Any other questions out there?
I've got one for Roger.
Yeah, go for it.
I'm curious to know, like, okay, so you find all of these background interactions that are happening and they're related with resistance. So is that more like adult plant resistance versus seedlings? Like, what is the relationship that's going on there? I mean, is some of what we're seeing in adult, plant resistance due to these like more complex networks that are underneath versus, seedling resistance being more simplified, kind of one-to-one argine kind of a, relationship?
That's a good question. The libraries were made out of a time course from 0 to 48 hours after inoculation from seedlings. And that encompasses almost all the proteins in barley in the leaf. But whether or not, well, that's what I'm saying. When we figure out the networks and we can tie the addresses to like a QTL in adult plants, I think that's what maybe close to what you're asking. But right now we don't have an answer to that question.
I mean,
it would probably be a good prediction that some of the end results would tie into seedling resistance, but not adult plant, and vice versa.
yeah, I'm almost wondering, like, could you just repeat the exact experiment in adults, and then you could look at what is kind of shared between them versus what is unique to adults versus seedlings, right? In the
That would
exact
be
same
possible
germplasm.
if you had a whole lab in a lot of years.
All right, good answer.
So that presentation I gave was 5 years worth of work. I
Wow.
started with
Okay.
Mitch Elmore. He's as postdoc in the lab. He's now a PI at Served Disease Lab, working on Fusarium, and then another postdoc and another graduate student and another graduate student, and yeah. So, and now we're just trying to focus and produce the network to the community so they can look at all the have all the addresses and apply it to their work as well.
Cool. Well, thank you.
Sure, thanks.
All right, any other questions?
I do have one question for Nate.
Yeah, why don't you go ahead?
Nate, is XTT specific to barley, or does it infect other cereal crops too, or wild grasses?
Repeat that. Yeah, this is Nate. I switched to Jules' computer.
Okay, go ahead.
I think
I'm sorry,
you might
could
need
you
to
repeat?
the question.
Okay, is XTT specific to barley, or does it infect other small grain cereals, including wild wheat varieties or species?
Yeah, so XTT seems to be specific to, that's the path of our translucence, seems to be specific to the Hordeum genus, at least. But it does infect also wild hordeum varieties, and some strains have been isolated from some other species, some other hordeum species. But it does seem to be very, to my knowledge, very specific to the hordeum genus.
Yeah, the reasons that I'm asking is I was doing some inoculation tests and I observed that oats might be a host to it because I have seen some inoculationists in our program and have seen oat seems to be susceptible, but just wanted to know if you have got any information on that or not.
Yeah, that's really interesting. I don't think I've seen documentation of pathovar translucence infecting oat, but I wouldn't rule it out as a possibility. It seems like they can be a little bit more fluid than the pathovar typing system initially, the pathotyping system initially allowed for. There's also some strains like I believe cerealis, which is a different pathovar, can infect oat as well as barley, but I have to double check.
Okay, maybe we will communicate and do some work together. Okay, thanks.
Yeah, definitely.
Okay.
All right, well, good news. Juliet is with us. She was tied up with the cereal school that she was working with. So I am going to go ahead and share my screen again. Has everyone seen that University of Idaho slide now, it looks like? All right. And Juliet, if you want to come off mute.
I am.
Awesome, thank you. And you just let me know when you want me to click through the slides, okay?
Sure. I just thought it would be good to give an update on the cereal cyst nematode stuff that we've been working on for several years. Pretty much the cereal cyst nematode distribution. We're still working on a whole bunch of different things associated with not only cereal cyst nematode, but other nematode issues. So go ahead and change the slide. What this slide is, it's an older slide that we've had in our back pocket from a long association with Dr. Dick Smiley. This was a historical distribution of cereal cyst nematode throughout the different areas. We also have the identified areas of H. aveni and H. philipjevi. So Heterodora aveni has been widely known to be throughout this area many, many years. And the philipjevi was discovered in a couple of areas in Oregon and Washington, and also now in eastern Idaho, up into the Ashton area. We've got some molecular identification of philipjevi associated with some work done by Poria Insafi, who was my graduate student and now is a postdoc with my program. Some of the other things that we are doing will be additional work we're pursuing with a new graduate student, and that would be Nadezhda Porras, or we call her Nadine. So the distribution, Colorado, Northern California, Washington, Oregon, Montana, and Idaho has been well documented. So would you change to the next slide, please? So just if you're not familiar with what cereal cyst nematode looks like areas of poor growing crop. Oftentimes people think that it's in this, why is my crop not looking quite well? Looks a little bit like nitrogen deficiency. The plants are not tillered as well. They look like they're suffering from drought damage. Then you start digging up and see the typical damage associated with the infections on the base of the root, multiple proliferation of the root system at and below the point of nematode infection or penetration in the stem, where they initiate the nurse cell development. And oftentimes we can see, and it makes sense, you see multiple nematodes developing at one site. And when you have a nematode cyst in the spring, when it germinates or when it hatches, the nematodes hatch from the eggs in the cyst, they are located pretty close to each other. And so it makes sense that you see the penetration of multiple nematodes at one close point together. But you got to be very gentle when you're digging your plants to see that very typical proliferation of the root system or a knotting type thing. Go ahead for the next slide, please. And this is symptoms, looks like nitrogen deficiency or some other nutrient type deficiency associated with plant growth. Very noticeable earlier in this season, and then as the season goes on, the plants tend to mature quickly. So in that area where you see the lower height, plant height, it'll also mature much more quickly in that area. So next slide. So what we've seen, this is just a quick slide to show some tremendous differences in the height and tillering of the plants. This is an old slide again, 2011, but we've been consistently doing work where we've seen 30% higher grain yield when you protect the plant against the cereal cyst nematode. And in some cases, it's a lot more than that. We've got areas where you have 10, 20 years of consistent barley growth, and you end up with huge yield deficits. And I'm talking 50 bushels of, say, spring wheat under irrigation when you have high levels of the cereal cyst nematode under irrigation. So the impacts are huge and widespread. In our upper elevation areas in sandier soils, that's where the most damage is, where you have crops that are, the crop rotation is limited. So go ahead to the next slide. So we've been screening for resistance to wheat and barley. I just put up the barley resistance results here, separated by malt and feed and food varieties. And what we've been doing is counting the average number of cereal cyst nematodes per 5 grams of root, and average number of cereal cyst nematodes per root. We get a lot of variability associated with this methodology because we are digging the plants out at a very specific growth stage when the female white cysts are visible. And the amount of variability is such that we have a proposal to modify the method associated with sampling to try to reduce the variability associated with the sampling methodology. If you can imagine going out to a field and sampling 5 plants per plot with a shovel, that sampling method will vary depending upon the amount of moisture in the soil, all sorts of other variabilities, how big your shovel is, who's shoveling what plot. And when you're doing a lot of work, you have multiple people often taking the samples, and you end up with too much variability to get a really good estimate on resistance and susceptibility. But even with that, you can definitely see on the left, AAC Prairie, which is a Canadian line, more recently being tested in our extension variety trials for yield and adaptability. And a Canadian line likely doesn't have the extent of resistance to a cereal cyst nematode in our environment. ABI Voyager is another example of one that's usually very susceptible to cereal cyst nematode. We know that LCS Odyssey has resistance to cereal cyst nematode, and luckily we do see that in our testing as well. So that's just an example of the multiple varieties that we've been testing. And then if you look at the feed and food lines, again, we still have a widespread variability associated with resistance and susceptibility. So our additional work, working with our new graduate student, is modifying our sampling protocol to try to get them a little bit more accurate reading, a little bit more standardized sample volume, and a little bit more standardized root sampling to get a better procedure. In addition to the graduate student doing work on this, we are also doing an overall survey of plant parasitic nematodes throughout the region to look at and to pinpoint the distribution of a lot of our cereal cyst nematodes, especially trying to pinpoint the exact distribution of the Heterodera philipjevi, which seems to be pretty widespread in the Ashton area. That's important because the resistance that we see for cereal cyst nematode the damage caused by Avini is going to be different mechanisms and different resistance genes associated with Philip Jevy. So I would say probably 99% of our cereal cyst nematodes are Avini. And we have tested this not only by the actual morphometric characteristics of the Heterodera species, but also molecular. So we've been looking at both types to characterize this Heterodera associated with small grains. And again, with our barley, it can be quite damaging, significantly damaging in the amount of yield obtained from our different variety trials and our different production regions. So that's real quick in a nutshell what we've been doing with our nematode populations in barley. So I guess at this point, if you have any additional questions, we can go over that.
Yeah, does anybody have any questions for Juliet?
Hi, Juliet, I have a question. Can you hear me?
Yes.
Yes, I can.
So I know that you said that y'all are working right now to modify some of your sampling measures, but have you considered taking soil samples at the end of the growing season and maybe comparing that to the beginning to see how much reproduction these nematodes were able to do over the season?
Yes, we have done that. And over multiple years in the past, we've done that a lot. If you're doing a screening nursery, when you have, and this is just a small sampling of the amount of varieties that we've actually tested, and doing a before and after sampling is a little bit difficult. But when you're doing a little bit more specific sampling, and we do a pre-plant and a pre-plot and then per plot sampling after. So you actually do get an estimate of the amount of reproduction and growth of that plant, of the nematodes in soil. So yes, we do.
Okay. Yeah, I mean, that is a lot of samples to take, but.
Yeah, so you're looking in this case, because of the soil variability with the population dynamics of nematodes, you're often looking not at 1 or 2 reps, you're looking at 4 to 6 reps, 4 minimum, 6 reps preferred. So when you go to a little bit more directed study, when you're looking at specific reproduction methods and stuff, we'll do 6+ reps, and we'll limit the number of varieties that you test, because otherwise we literally don't have enough people that we can hire to do the work.
Right. All right. Awesome. Thank you.
Sex reversal too. Poria and Safi is actually online, which is excellent. And why don't you pop in and talk about the sex reversal?
Hi, just a quick note or add what Juliet said. There is a strategy in cereal cyst nematode and some Melodogyne too, that when the plant variety is resistant or the situation is not good, nematodes that are genetically female go through a sex reversal and turn to male, stop feeding and leave the root. So pre-plant population and post-plant population, there is an issue. Well, the hatching is temperature regulated, so you always have difficulties in identification of the juveniles in the soil. So when you do the soil sampling only pre-planting, you have the estimate of the nematode from the cyst only, because you cannot identify the juveniles at that stage from soil samples. And the other one, well, you have the cyst, inside is the egg. You don't know which one is the new, which one is the old. So if you want to check the differences in the population, it's always the problem that you don't know which egg is viable. So you include in the countings which one is not. So it's there is some issues with the old technique pre and post-plant population differences for evaluating the population density. So we are considering the DNA extraction from soil and plant material. So it will be DNA quantification included in the new protocol as well. Thank you.
Excellent. Well, thank you so much for the team from the U of I for presenting today on this. Thanks, Juliet, for sneaking away to come with us today. And thanks to everyone that made presentations. I know this is just a very small snapshot of all of the amazing work that's happening right now, but in lieu of having a whole day forum, which would be quite painful on Zoom, I just wanted to do some of these kind of quick snippets, and hopefully, as I said, in 2026, for our next Barley Improvement Conference, we'll have a much more expanded BPI forum in person where we can really dig into the great work that you guys are all doing. So I'm going to switch gears now and talk actually about what we've been spending BPI money on and what pests we've been focusing on. Oh, forgot your last slide here, Juliet, but I think that was what you guys all just talked about there. So did you have anything else to add? Yeah, okay.
Not really.
All righty. And then I'm just gonna slide through these since we had that Idaho one first. Sorry, guys. That's a real lightning presentation there. All right. Okay, so if you've already gotten into Slido, I've got another poll. If you haven't been into Slido yet and want to participate, go ahead and scan that QR code or go and open a web browser to slido.com and enter that number. Just the question that I want to ask before we kind of dive into the data is, what do you perceive in the seat that you're sitting in right now, the program that you're working in, what do you perceive to be the greatest pest, insect, or disease to barley right now? Doesn't have to be even what you're working on, but what do you perceive to be the biggest challenge to barley? I'll give you guys just a few minutes to enter that. And this is a word cloud, so the more something gets typed in, the larger it will be. And it's interesting, last year when we did this, I said we could only say things that were not fusarium head blight, but this year I decided to put that in there because obviously that's huge, tremendous concern still. We have a whole initiative devoted to that work. And so this year I was kind of interested to see how that might stack up against some of the other pest threats that we're dealing with. Give it a few more seconds for folks if you want to drop in your idea. There's the ultimate waffle out there, FHB and BLS. I'm sure maybe a lot of people would have said that too. All right, we'll go ahead and close this one out. So bacterial leaf streak actually is coming out ahead as the winner. And for those of you that maybe were focusing just on the BPI pest, perhaps not thinking about FHB, that it certainly was reigning kind of superior to the others. So thanks for doing that, because what I want to now show you, and you probably, if you looked at the BPI annual report, already saw this, but I wanted to dive into actually how those funds are getting spent and what we are focusing our efforts on. So here is the distribution of BPI funding right now. And now keep in mind, this was the reporting work that was done that only included the first $3 million. So we are not fully funded. We don't even have that $4 million that's tentatively in the budget yet. So this is work that's just been accomplished with the $3 million annual appropriation over the past now 3 seasons. Stripe rust is coming out as the leader in terms of funds received. Then we have number 2, bacterial leaf streak, which was in our previous slide, the #1 concern, which I would say too, I should have looked, but I think this bumped up maybe a couple places. So I do think we're devoting more money to BLS than we were even last year. And then we've got powdery mildew, stem rust, net blotch, leaf rust, yellow dwarf virus, and then one of the aphids. And then 16% of that funding are the others. And I will go to the next slide and show you what those others are, but just wanted everyone to kind of get a good look at this and let it soak in. And then go to the next slide. So these are those others that makes up about 16% of the funding. Got green bug, Russian wheat aphid, spot blotch, nematodes, scald, wheat stem sawfly, loose smut, and then also, as I mentioned, the support towards grain genes. So that is how the funding is distributed. And then I also wanted to highlight the initiative did identify as being targets, but at least in last year's report, we did not have anyone reporting that they had done any work in these areas. Predominantly.
Can I add a comment?
Yes, please.
Ashley?
Mhm.
A lot of the pest orphans that you see here, I mean, I'm doing work on wireworms and seed treatments and things like that. It just seems to be a constant low problem. We're not focused on it. The Hanchin barley mealybug is so dependent on weather to actually even pop up and be visible. It is really not something I don't believe that anybody has been able to consistently get an infected plant or a system down to get constant infection. Very localized into the Intermountain West.
Thanks for that context. And I certainly know that just because these were landed on a list once upon a time doesn't mean they necessarily need to be our focus now. And I do know that even on the disease end, some of these are a bit challenging. We're not seeing, repeated priority kind of disease pressure. I will just, go back. I've got another slide that we'll get to, but thinking through this, I just wanted you guys to all see kind of now what the distribution was. I've got a slide, a couple down the road that will actually kind of lay these all out, because I do think it's good to have a discussion on if there are some things that aren't getting funding pushed towards that we should be, perhaps even thinking about those priorities. And that's what then got me ahead to this next slide. So I put, just for purposes of this, this poll, I put what those top 8 diseases were in that pie chart, and ordered them 1 through 8. And in Slido, you can actually, that's popping up on your screen, you can use your finger, if you're on your phone, to actually hold down these and drag them and try to put them in order to where you think the priorities need to be. Again, there's no right or wrong answers here, just curiosity again from our researchers. If we need to be moving some of these things up in terms of priority, do we need to be thinking more collaboratively about how we can prioritize funding for some of these? So if you're on Slido and want to respond to this, we'll take a few seconds to let people do that. And then I believe it's the next slide where we'll put them up all on the screen and have just a general conversation about this. Oops, sorry. Oh, shoot. Again, trying to run the Zoom and monitor all the things. I'm not doing a great job at today, so sorry. Just as you guys are responding, I saw Kelly, your message about Doe working on the hedgehog grain aphid, and I know she does. She didn't report that she used BPI funding for that work this year. So, but I do know and recognize that there's plenty of other work happening behind the scenes that just, aren't really leveraging BPI funding to do that work. So trust that I recognize that too. All right, just give it a couple more seconds here. All right, so what I'm seeing, we've got bacterial leaf streak, perhaps coming out as the priority with stripe rust following, and then the powdery mildew. A little interested, and feel free to just chime in. Are we having more concerns about powdery mildew, perhaps because of an increased interest in winter barley, or are we just seeing powdery mildew be a problem universally? Anyone have any thoughts on that?
I have a thought, but
Mhm.
this survey seems to be a little bit biased by just who's contributing. And everybody has a conflict of interest.
That's why we're just doing it
But-
for fun, Roger.
But
Nobody's
the question
publishing.
is, some of these diseases like powdery mildew, is a model, kind of a model obligate biotroph. It's much simpler than rust, but rust itself actually has all kinds of things that can be learned about the rust, about all rust, stem rust, leaf rust, stripe rust. I mean, all the diseases contribute something to host pathogen biology, and some are uncommon and some are unique. So that's why that's a separate kind of topic than what's actually the problem right now in the field. Because we might come out with something now that it has an impact 5 years from now or 5 years before now. And so the research engine should keep going and keep contributing to solve problems as we go. But I don't think you can respond to something in a year by year.
Oh, no, I certainly agree with that. Really, the point of this exercise is if there are emerging diseases that we're not addressing or perhaps not allocating enough resources to, I just want to make sure that we're getting out ahead of that. I think oftentimes we're affirming the work that we're already doing, and I think that's great. And I know that you all have stakeholders that you are accountable to and making sure that the work you're doing is relevant to them. So I don't question that at all. I just, like I said, want to be strategic in thinking ahead. And if are there emerging diseases, let's say BLS was actually number 8 at the bottom of the list in terms of getting funding, I think that's something we might want to respond to. We're not seeing that. We're actually pretty much in alignment with how the funding is getting spent each year. So I think that's great. But I think that there's only a few opportunities where we get to have these group conversations. And so I just wanted to take advantage of that today. So really appreciate the dialogue on that, though. Jonathan, why don't you go ahead?
Just a quick question. In the Barley Pest Initiative, is there opportunity to allocate funds for those emerging problems, or is it, and what's the future direction for this so that we respond to emerging needs or industry problems? Is there going to be actual applications for projects for funding rather than allocating money every year to a lab based on that?
Yeah, that is a great question. So the way the Barley Pest Initiative was structured is that there were a list of these pests, over 20, and then there were programs, so to speak, whether university programs or programs at ARS sites that were essentially assigned to work on these pests. Now, I have a big spreadsheet where I take all of the data that gets reported, and I actually do track what pests you're working on, and is that what we said we were going to work on, and if not, which ones, do we still need to work on? And that's what kind of helps bring this presentation together. It's kind of a long-winded way of saying that the way the program is set now, it's pretty set in stone with the pests that we have. To make any changes is going to be rather difficult in terms of how much money is going to a certain location. I believe that the only way to make any adjustments would to increase the appropriation on the initiative that we have to Congress right now, which I don't think is going to be a very successful venture. Now, that's not to say it can't be done and that we couldn't look for alternative funding elsewhere, but it just might mean that we need to reallocate or also come up with creative ways to collaborate across locations, especially with looking at university and ARS sites that could potentially work on things together. and just be a little bit more efficient with the resources. So yeah, there's not really a great way, at least in our kind of congressional directive, to make many changes to what we're doing already. And that is just the nature of how this initiative was developed by Mike Davis and many of the partners on that initially. So this is something I inherited it. And if I'm misspeaking, for those of you that maybe were there at the inception, please let me know. But that's the way that I understand it now. And speaking with folks more at the administrator level within ARS, it's the way that they understand that they're supposed to essentially, administer and then distribute the funds too. So.
Yeah, I'm just concerned if there's a big outbreak of a new emerging threat, like how would we be prepared with funding and also variety choice use, right? Like if there's a huge shift to winter barley, but everybody is focusing on spring, I mean, we could be in trouble if even the industry shifting, right? So I just, yeah, like how can we be nimble if there are emerging threats?
So one thing that I would say, and then I'd be open to other people providing comments on this, is that I think it's really important, especially your perspective from wherever you're at. So Jonathan in Ohio or Juliet in Idaho, when you're starting to see things that are of concern to you, that you perhaps don't feel you have the resources to address, it's never too early to come to AMBA and the National Barley Improvement Committee to bring these ideas forward, to make sure that we can be a partner with you in identifying resources to tackle those things. One option is, of course, through our AMBA grant program, which I recognize is a very modest grant program, but if there's a way that we can take those funds and leverage additional funding elsewhere, we would love to help be a partner in that. So, my message is if you're seeing emerging pests that you do not feel are encompassed by the BPI, or you feel you have the resources to tackle through the BPI, you definitely need to let us know. Don't stay in a silo on that. We want to make sure that we, as your industry partner, understand that, because we can help try to fundraise to make sure that those issues can be addressed. And I don't know if anybody else has any additional thoughts or comments on that.
I kind of have a question.
Yeah, Jamie.
So if we notice something, so every year there's a renewal. So the people that are getting money, every year there's a renewal. And we're, at least so far we've been adding to it. If we saw something emerging or had a new idea, could we, in that renewal process, shift some of it to it doesn't include a new researcher necessarily, unless we're willing to share some of the funding. So for example, we don't have any soil-borne fungal work, and I have a collaborator here at MSU that's interested in starting to screen material, and so I'm going to share a little bit of the BPI money with him to see how that could go this summer. And I sort of thought that we had that flexibility, that as researchers we could kind of add that on if we wanted to. I don't know if that's true or not, and so if I'm not supposed to do that, then you should tell me to not do that.
No, that is definitely. So like any of the pests that are on the screen right now, and I can't remember if there's 21 here or some odd number like that. Any of the pests that are on the screen right now, and if you're already a part of the collaborative network, you have the freedom and flexibility to utilize the funds at your site to address those. So absolutely what you're saying, Jamie, is 100% okay. And
so then I guess another thing just real quick to say is that we recognize this as a problem, and of the lack of flexibility is a problem. And so with the resiliency initiative, which is our newer ask, we've included in that this possibility of I don't know what we're calling it, not competitive grants, but more flexible money. We're including more flexible money in that once it's fully funded. So each year, if there's a new issue or somebody comes up with a great new idea, there's going to be money to address that, hopefully. And maybe eventually we could get that added on to with the BPI. Like maybe we could eventually fix this issue with the BPI as well. That
Yeah.
would be my hope.
And I am going to talk about the new initiative here at the end, and I want to address that because that was going to be my other idea to really kind of tie on to Jonathan's comment. And so let me get to that. I just wanted to see if anyone had any additional comments on the pests that we are working on. Are there emerging pests that are not on this list yet that we should be thinking about?
Well,
Any
Ashley, I had
comments?
a quick
Yeah.
question. Is there any interest in industry actually funding something like, you know, new initiatives? Or is that going through AMBA?
Right. So when we think about the initiatives, I think of like the Wheat and Barley Scab Initiative, the Barley Pest Initiative, and then our new Resilient Barley Initiative. Those are all congressionally funded initiatives. I think industry certainly has interest in funding work, but right now they're choosing that vehicle to be the AMBA grant program. But what I will say is that there are some of our industry partners that actually give above and beyond their membership dues into our grant program. And then they essentially earmark what research programs they want to invest in. And a good example of this, and I only repeat it because it's one that I benefited from when I was at Michigan State University, is that each year the Michigan Brewers Guild gave a very modest amount of money to AMBA, then to channel through the AMBA grant program to fund our research. And those models exist, and we are very happy to take additional industry dollars. And AMBA is actually rolling out in probably the next just few months a new membership category that will make industry investment and research a little bit easier, because right now our membership is only open to maltsters, brewers, or distillers. We are going to open up an affiliate category. So if you have an industry member that is interested in funding research, so if you've got a stakeholder that you've been working with, they can then start a membership kind of with AMBA and then be able to route their membership dues through our grant program. And I recognize that they could probably just write a check to these research programs directly, but thinking very strategically and trying to use, be really effective and efficient with the dollars, we do suggest people to work with the AMBA grant program if at all possible. And so that would just be another example of ways to get things funded through industry.
I was actually directing that question that when I said new initiative, I meant like Jamie was talking about, you know, new emerging diseases, if they were willing to put money, like, and I think you explained it pretty thoroughly about you're going through AMBA to do that.
Yeah, so that would be the vehicle, at least that AMBA is facilitating for industry to be able to do that. But I'll since it was mentioned and we're coming up towards the end of our time together, I will just give a brief overview of the next initiative. This does not replace the BPI. So the BPI will still exist. We will continue to fundraise for it, but we are taking to Congress next month this new initiative, the Resilient Barley Initiative, which is going to be a very similar collaboration with ARS and our university researchers. Many similar states, some new, some maybe that aren't going to be participating that are with the BPI. But this initiative, the total financial ask is actually going to be $8 million. And we recognize that it's going to take quite a while to fundraise that. But to look at the funding breakdown, there's about $2.5 million to support our breeding programs, $2.5 million to support management trial work, which is relatively new and different compared to BPI, where a majority of the funds are all going to breeding. And then we have about $500,000 annually allocated to actually fund a person. And we don't know who this person is, where they will be, and exactly what their job title will be. But we recognize that the BPI already has become this huge effort, not as big or significant as the Wheat and Barley Scab Initiative. But if you think around all of the resources and the capacity and staff that work on that, We're just kind of a small fraction of that, but we've got no designated staff to work on the BPI. And to make sure that these coordinated efforts happen, to make sure we're on top of emerging diseases, and also thinking about how we can leverage our dollars to go after additional funds. So with the Resilient Barley Initiative, we're really hoping to have someone in place that can actually work on the RBI, but perhaps then also help with some of the coordinated efforts around the BPI. And so that person is going to be a priority as we take this initiative to Congress, to get someone in place to help with that. And then what Jamie mentioned already is that at the end, the final 2.5 million of that 8 would be highly flexible dollars to go after the things we need to work on that maybe we didn't think of in 2023, but in 2024 or 2030 or 2040, all of a sudden is an issue. And so similar to how the Wheat and Barley Scab Initiative provides that external funding into those programs, we will operate, and again, I don't even want to call it a grant because I don't know what that's going to look like, but operate a program where people have emerging needs and we have more flexible funds to help support that. So just looking at what our targets will be in this initiative, when we think about on the breeding side, the 4 areas that we're focusing on are temperature stress, moisture stress, thinking about pre-harvest sprout, because that has become a major quality concern, especially in the malt industry. And then also really trying to focus on winter hardiness so that we have some new winter barley varieties. I should mention that all of this work is meant to focus on spring and winter, of course, except with the winter hardiness piece, but this is not an initiative just for winter barley. I think that perhaps maybe that's been misunderstood. We definitely want to be working on things like drought and heat tolerance in spring barleys, a major issue that we're having in Montana, for example. And so these kind of 4 areas are really focus on the breeding effort. And then when we think about the management work, we are looking at cropping system trials. And a lot of this is going to be, involving winter barley and just trying to propose new strategies on how we get those crop in the rotation. But down when we're thinking about management trials, how can we be more efficient with our inputs like water and nutrients? That's going to be across all barley types. And then also doing a better job at taking all of this research and informing recommendations. And that's where the person that gets hired through this initiative is really going to be working, is making sure we're getting this translated back out to the field in a more applied fashion. So that's the Resilient Barley Initiative. And I'm really excited, as I said, to take that to Congress. And I'm hoping that those last $2.5 million we'll be able to use on emerging threats. The Resilient Barley Initiative is really focusing on the fact that we have abiotic stressors in the system, predominantly related to climate change. And I think that many of you are very aware that a changing climate has actually impacted the pest threats that you're dealing with. And so there's no reason why, if there wasn't an emerging pest issue that we're not able to tackle with the BPI, why we couldn't try to route those concerns through the RBI. So I want to leave a little bit of time here at the end for any additional questions. So with that, I'm going to go ahead and stop sharing my screen and open it up to everyone. So questions on what was presented related to the Barley Pest Initiative, questions about what's ahead in the RBI, just open it up for everyone.
Actually, I just talked to Dr. Andrew Friskoff, and he reminded us that our #1 pest is better malt contracts.
Oh, perhaps true, but I'm sitting here as a representation
Out
of
of the
the
scope.
industry, so I cannot comment.
Out of the scope, I gotcha.
Additional comments? All right, well, that's not the note we can end it on, Thomas. Don't you have anything better to say than that?
I have a
Our number,
Roger.
our foliar pathogens of interest are bacterial leaf streak and the net form version of net form net blotch complex. So we are pretty well in line with what we see on the screen. We don't have really much rust, but we do understand that other states are getting a lot of rust and need that research.
Great, thank you. And I just wanted to comment, I meant to say this earlier, when I do go through all of that, the reports that you guys send in to me, it is amazing to see how much collaboration is happening across programs, even if it's not local. I mean, I know that we've got great already established partnerships between universities and ARS sites that are sometimes even co-located, but I'm seeing many of you reaching much farther beyond that. So I just want to tell you all how much I appreciate that. I would love to do some network mapping sometime with BPI and actually show how interrelated a lot of this work is. But I know that sometimes it takes coming together like this to make sure those things happen. So I really, again, appreciate everyone spending some time together so that we can share ideas and what we're working on.
And I don't remember if there was an output on your document, but it should be, if it's not there, that some BPI funds had gone in to support the bacterial leaf streak research initiative, which is both wheat and barley. But on the barley side, we've had fostered collaboration on it, and produced the bacterial leaf streak research initiative out of it. So it was we thank at each of the meetings starting in Fargo, we thanked AMBA and BPI.
Awesome. Well, thank you. And I was actually hoping in the next reporting season to add a question to the survey about how have you leveraged these funds for other work and started some of these new initiatives. So I definitely hope to capture some of that next year. I know that only a few years in for folks that was maybe a little too early to ask that we'd get much feedback, but it's really good to hear that those things are happening already. And I do hope in the next questionnaire round for next summer that we will be asking and hopefully hearing some of those success stories. So thank you. Well, it is 2:30, so I am going to go ahead and stop the recording and just again want to say thank you to everyone. I can stay on for a bit longer if anyone wants to chat, so I will do that. But again, thank you. Have a wonderful rest of your February. For those of you that I'll see in DC next month, see you soon. All right, take care.
Thank you, Ashley.